This is a cartoon of what the virus looks like, but it's a really accurate cartoon. So it's based on actual science and experiments that have worked out what the structure of the virus is. And you can see the spike protein, which is the pink thing on the surface. So this is showing the surface of the virus. It's kind of cut in half on the right hand side. And the spike protein is what gives the virus its name, coronavirus, corona as in crown, because of these spikes that look like a crown. And the spike protein many of you have heard about, because it is the part of the virus that the vaccines in particular try to target. So the virus has this just this envelope of lipids and proteins, and the spike is a protein on it. And then inside this casing, it has its genome, its RNA, and that's what encodes all its different proteins, and that's what gives that's what replicates to give you more viruses. So when we're thinking about our immune response, this is just a very simple picture. Different terms that you've heard about. I mentioned that my interest is innate immunity. That's the early part of the immune response, and then adaptive immunity is the part of the immune response that's more like the memory that develops as a response to our immune system's learning about the virus. So on the left-hand side here, if you're infected with SARS-CoV-2, so SARS-CoV-2 is the name of the virus that causes COVID-19, in the same way that HIV is the name of the virus that causes AIDS. So COVID-19 isn't the virus, that's the disease, and SARS-CoV-2 is the virus. So if you're infected with SARS-CoV-2, your body, if everything's going well, will develop antibodies and it'll develop T cells that will then fight the virus. And this will happen, a couple of days after you get infected. And if that's a very successful immune response, that might clear a certain virus. And then when you recover, you have then these antibodies still in your system, because these have been left over. These are now been made by cells, and you have your T cells, but also you have B cells as well, which are memory B cells. And so the reason that we give vaccines is to try to get this recovered response without people having to be infected in the virus in the first place. Because if we show bits of the virus to the immune system, then that actually activates the adaptive response. And that's why you've heard lots about antibodies and less actually about T cells. So with a vaccine, what I'm saying is that the purpose of the vaccine is to show a part of the virus to the immune system in a safe way. And it's sort of trying to mimic the initial infection, but you do it in a way that is using bits of the virus that aren't infectious, so you don't get all the different pathologies that result from being infected with the virus. And therefore you're kind of in this state after vaccination, that it's like your immune system has seen the virus and it's ready, primed to fight the virus better. So this picture here, I'll just spend a little bit of time, because hopefully this will help people understand the different vaccines. It's a little bit complicated, but again, here is the normal virus. And what immunologists and vaccinologists do is they work out ways to safely show parts of the virus to the immune system. So when you get injected with a vaccine, it's a certain way to show a part of the virus to the immune system. And the main thing that we show to the immune system for these vaccines is the spike protein. So we basically show the spike protein to the immune system. The immune system gets tricked into thinking that there's a real coronavirus there, and it develops all the different antibodies and T cells that will then help you to combat the virus. And the vaccines are different in the way that they try to show the protein to the immune system. So, for example, number one there, the RNA vaccines you've heard about from Pfizer and Moderna, they have a piece of RNA that codes for the spike protein. And when that piece of RNA gets inside your cells, all our own cellular machinery can produce the spike protein, which then turns on an immune response. So one way to show the spike protein to the immune system is to use these RNA vaccines. Another vaccine is where you, in fact, just use the protein itself. That's harder to make and more expensive, but you can make the protein in the lab, and you can then just show that protein directly to the immune system. And then also a third way is that you can use a different kind of virus, a safe virus, to produce the spike protein and show that to the immune system. And that really is the basis of all the different vaccines. So if we just put up this here, there's a lot of information here, but let me, there we go, yeah. So hopefully you can see that. So this here is really just the different types of vaccines that are currently being developed or have been approved based on those different technologies. And here, so in the first one here, this is the RNA vaccine. So this is what Pfizer and Moderna use. That's where we deliver a piece of RNA into the body in the injection, surrounded by a lipid coat, and that then gets inside our cells, and it then expresses it, it decodes the RNA and expresses the spike protein, and this protein then alerts the immune system to turn on and to fight the spike protein. And so an example of this is the Moderna and the Pfizer vaccine. There's different types, so I just referred, there's kind of 5 different main strategies, but the 3 that you will have heard of are, well, the 2 mainly is the RNA vaccine and also then viral vectors. So this is where we use a different type of virus, and it's often what's called an adenovirus, and an adenovirus is also called a DNA virus because its genetic material is made up of DNA, and so adenoviruses can be used to express the spike protein and show that to the immune system. And so AstraZeneca, the Oxford AstraZeneca one, uses a chimpanzee adenovirus, which is safe and doesn't cause any illness in people normally. And Johnson & Johnson, it's the same kind of approach, but they use a different type of adenovirus. So the RNA vaccines are Moderna and Pfizer, and they're basically presenting some RNA to our body that then gets decoded into the spike protein. And these Johnson & Johnson and the AstraZeneca Oxford viruses, these are live adenoviruses which are safe, which express the spike protein, and they show that to the immune system. And the one you'll also hear about a bit later in the year is the Novavax virus, and Novavax is the pure protein itself, and you are presenting the pure protein to the immune system. So it was useful to show those slides because there's lots of questions that have come in about the different vaccines and comparing them. And so again, just let me summarize it by looking at this protein. What we're saying is the viral protein gets presented to the immune system in all these different technically different ways, and the goal is to get the formation of antibodies against SARS-CoV-2. So I'm going to stop sharing my screen now. Actually, no, maybe I'll leave that up. I can see the chat as well. I can't. So some of the questions, let me go through that are related to this, which this picture might help with. So I wanted to first of all show that, and then, so first of all I'll address the question about the fetal cells. So how are these vaccines made, and does it actually evolve fetal cells? And so the answer is yes, sort of. So many of the vaccines that are developed, including vaccines for rubella, chickenpox, shingles, hepatitis A, many of these use cells which are called HEK293 cells. And what these are is these are cells whose ancestral cell came from an aborted fetus in 1972. So what's happening here is that there was an elective abortion back in 1972, and people took some cells from that fetus and started growing them in the lab, and found that those cells just kept growing. And so they've become very useful because millions of cell replications later, in 2021, these cells are still growing. And they're growing all over the world in all different labs, and so the actual physical material from that aborted fetus would no longer be actually part of these cells because they've multiplied so many times. But it is true that the cells that are used in developing vaccines originally came from a cell in an aborted fetus. And so as I said, these are the HEK293 cells, and so specifically the viral vector vaccines from AstraZeneca and Johnson & Johnson, they would have used these HEK293 cells because you have to use cells to grow the viruses. Whereas the RNA vaccine, the Moderna and Pfizer one, they would not have used these HEK293 cells to grow. So it's not true that the vaccines contain fetal cells or fetal tissue, but it is true that part of the development process of some of the vaccines did involve using a cell that has an ancestor cell from a fetus 50 years ago. And so that's, and obviously some people would still have an ethical issue with using vaccines that are developed in that way. Personally, I wouldn't, but some people do, and that's fine. And the ethicists have different views on this, so that is a factor. I mean, it's interesting that the Pope has come out actually and said that, look, basically it would be better if we had other ways to make vaccines that didn't involve this, but for practicing Catholics, it is okay to take this vaccine. But let's hope that someday there's a better way to make these vaccines that doesn't actually involve having to use those cells. So that's a bit on the fetal cells. I don't know, Loxley, if you want to come in at all on that issue.
I think this is the core issue of many core problem of many Christians here. I don't want to spend too long about it or to suggest what personal decisions we should make. I just want to raise the point that, you know, we face ethical issues, ethical problems every day of this nature. And I would ask us to ask ourselves, why is it that some of them bother us and some don't? Because the core question really is, can we, are we able as Christians to use or make use of for good anything that comes from a morally debatable act? That's the core question, I think. And so just to name a few issues, you know, I mean, you know, things like mustard gas developed to kill people in the context of the world wars was on the basis of that we have chemotherapy. You know, questions like what are children, the children of the dispossessed in Africa to do or say about their neighbors who are descendants of those who dispossess them? Oftentimes they're both Christians, right? What are another good question is, which is, you know, what is